Thanks a lot, Grant. I'll start with a statement. I believe that Australia and New Zealand are world leaders in plant biosecurity research. And so I make that statement just on moving around, going to the Northern Hemisphere quite regularly, probably too much. But I really see, just in my area of diagnostics, how some of the things we do and develop over 5 or 10 years, other countries and regions pick up. And we see great examples of quality research through this CRC. So in talking about this project, I really wanted to spend the first 5 or 10 minutes building up to how we got there. And the three reasons I really wanted to do that was just to reaffirm to our funding bodies, just another example of how important continuity in research is. A reminder to the participants of the CRC just how lucky we are that we've had 13 years of research dollars in plant biosecurity. And probably to the younger scientists, if you get a good idea, stick at it, because you'll get an idea of how long it takes to actually get some research dollars to investigate it. So that's sort of where I was going. And to do that, I thought of Sir Isaac Newton when he said, we're standing on the shoulders of giants. And so some of the giants that's helped just in this particular project that I think have made a big impact. The first was a whole group of giants, I'd imagine, from around the world who in the 1980s must have started to think about free trade. And they came up with this SPS compliance agreement in 1995. There would have been representatives from Australia in that policy, but it is brilliant policy. And the catchphrase we use that we've all heard around that is not known to occur. And if we think about that in the 1995, in the 90s, when really biosecurity wasn't a term, this really helped to shape the way we think and safe trade based on science as evidence. The second giant that was really important to this project for us in Victoria was Peter Merriman, Dr. Peter Merriman. Peter, specifically with this project, he got money from the then HRDC in collaboration with the apple industry to do a contingency plan on fly blight in 1995. And in that, it was a book. My God, thank God we don't have to do that anymore, but it was an absolute book. But one of the chapters was a diagnostic protocol for amylovera. And when we got to 1997 with that incursion, we had that on the shelf. We had positive controls, we had primers, we had formula for media. So his vision before we had that incursion, to me, was one of the first examples of being prepared for an incursion. The second giant, again in Victoria, was Jane Moran, but she was certainly a big player in the first CRC in plant biosecurity. And from a Victorian point of view, she shaped a lot of our research during the 2000s, the late 90s, 2000s. I remember the day we did a budget for a Victorian contract, and the yearly budget was over $1 million, and we just looked at each other for about five minutes saying, well, we've never done that before, but Jane was very good with budgets. The next giant was Simon Curdy, and I think for us in this current CRC, he and the board and his team and all the participants in the first CRC had a lot to do with this current CRC we're in. And just remember the courage, I think, of continuing on for another year when we got the first rejection of this CRC. Absolute bravery. I always thought that was a strong decision, but that's why we're here 6 years on talking about those outputs. And the last giant down to our project, one of the great things about the CRC, and we talk about international collaboration, is our project leader, Jim Stack, who sadly couldn't be with us. He's just with some personal issues. But Jim, our project leader, my God, he taught us so much. And, our PhDs that hang off the side of this particular project, he thinks big, he gives you confidence, and he really helped us be bold in some of the questions we asked. But Newton was sitting under an apple tree when the apple obviously fell on his head. And so a very reliable source, Wikipedia, told me that this is the actual apple tree that he was sitting under when the apple fell on his head. And so I just wanted to link this back to our project because under extremely close inspection, I realised that this tree has no fire blight. So now this sort of relates to our project because one of our thought leadership papers was the new and emerging bacterial diseases, and this to me is more evidence to say that fire blight emerged from the Appalachian Mountains in the 18th century, 17th, 18th century, and it actually wasn't in the UK in 1676. So some of the ideas that come out, and so this is this concept. So in about 2003, Jane had this absolute brainwave about the ladder. She called it the ladder. And if we start down the bottom, we can see we're talking about if we've got something new, we're resolving etiology. And we're working all the way up to number 9, which is nationally accepted QA protocols. And isn't that interesting that in the early 2000s already we're thinking about standards and quality assurance, and now NADA is so important for our labs to run protocols, and we're now, industry and funding bodies are now starting to invest in proficiency testing to raise the competency of staff actually doing the tests. So again, that's sort of an example of how long things take. The other one was the Mobile Diagnostics unit. This is a classic. So we started mucking around with this in Victoria in the mid-noughties, and it was pretty amazing. So we had this sort of van and you had all the stuff in the back, including about 3 or 4 litres of ethidium bromide so you could stain your gels. I'm really glad we never got rear-ended because that would have been an extraordinary worry. But we did it though, and so we were amplifying RNA viruses in the field, and Simon got hold of this in, must have been 2008 or 9. He was thinking about the next CRC and he wondered whether this could be sort of a highlight of trying to make diagnostics mobile and into the field. And of course, one of our main drivers of this project we're talking about is in-field diagnostics. But anyway, back to some of the science that led to our project, and that was that incursion on the 3rd of May 1997, and yes, it was a Friday, always happens on a Friday. What happened here, and some people are bored with this, but what happened here is we got these bands up the top, and from a PCR perspective you think, yeah, that represents a Erwinia amyloid, the causal agent. But what we found out after about 2 or 3 years is that these bands actually represent a completely different bacteria. And so we made some false conclusions based on the interpretation of that molecular test. And the implications of that, it did influence trade within Australia, and then we nearly lost an export market into Japan because of that. So this sort of opened our eyes to thinking, wow, there's more, this spectrum of bacterial species that are out there is much, much greater than we thought. And so we started to think about more specific type tests. And so we went on, and in the first CRC we funded a PhD project, and it was about the application of genomics approach to develop a reliable diagnostic tool for EA. And what we have here, this circle, you'll see a few more of these, is based, the circle represents a bacterial genome, and each different colour represents a different strain of that species. And this PhD was Rachel's Mann's, and it set up the science for the strategy that we're going to talk about. In terms of how things change, I think we spent $23,000 in sequencing to do those 10 isolates just over 10 years ago for that project. So that was Jane's budgeting again, thank God. But the transition to now is that we can do bacterial genomes incredibly cheaply. We're getting down to around $100 a genome. So that's that technology shift. So it's much more achievable for us. But out of that came a strategy, and so I won't go through each of these steps. I'll do it later and more refined. But out of Rachel's thesis came this strategy about a genome-informed strategy to develop diagnostics, which is where we are today. So, to our project, deployment of validated genome-informed bacterial diagnostics. And PBCRC 2002 was 2012 to 15, and our current project 2156 is 15 to 18. It's a big team, and it's got that international collaboration involving 3 countries. Down the bottom right-hand corner is Murdoch Uni, who are with us in the first project 2002, but the 4 main players in 2156 is Kansas State, and Jim Stack is our leader, AgVic, where I'm from, and Rach in New South Wales, led by Tony Chapman and Grant Smith's team in plant food research. So our goals or aims for each of those projects in 2002 was to develop and validate laboratory diagnostic protocols for plant pathogenic bacteria, and 2156 was an extension of that, and we really wanted to see how we could transition these diagnostics into the field. It was a really big driver of our investment in this project. And behind that was an objective to increase national capability in plant bacteriology. So when we had that first PhD with Rach, there would have been probably 2.5, 3 FTEs of plant bacteriologists in the country at that time. But we knew that there was some very serious bacteria knocking on our door with respect to threats to our industries. So the science, genome-informed diagnostics. So I'll read down the words first and then go through that flow diagram for you. And for the way we try to describe it is we say the best way to identify new diagnostic targets is by comparing the genomes of both target and close relatives. So what we're after when we do a molecular test is some DNA or a sequence that's specific to our diagnostic target but doesn't exist anywhere else. We don't want those false positives. When we identify those regions that are unique, we design primers for our PCR, conventional qPCR or LAMP, for regions from different locations in the genome. And we really like the concept of trying to multiplex if we can. So in the one test, we hit 2 genes in the genome, so we need a plus to confirm the presence of our organism. And then critically, the last green, it's so obvious, it is so obvious, test multiplex assays against a range of endemic isolates and plant material. It's so obvious, but you cannot believe how few people do it. And so that validation in your local environment is critical. It takes time, it costs money, it's not very publishable, but in biosecurity is critical. So if we look up here, it's such an awkward thing to, I could go over the top of my head, but I'm not that clever. So up here, this represents Rachel's 10 genomes. We identified a core genome. With that core genome, we compared it to these other Erwinia species, Tasmaniensis, Pyrifoliae, bleeding. So they're close related, so they're most likely to have genes similar to Amylovera. And when we look at those, we subtract those genes that are in this core to identify our potential targets. When we've got our potential targets, we then go onto this global database we call NCBI, where there's lots of bacterial sequences on there, and we blast our potential targets to all that sequence. And if we get hits, we take them away. The issue with bacteria, and we're starting to see a little bit with fungi, and I heard a talk the other day about between fungi and plants, is this exchange of genetic material we call horizontal gene transfer. And so genes can move very, very easily. So that's why it's important to compare to that global database. Once we've done that, we analyse the specific targets. So if they're specific to Amylovera, we still want to make sure they're somehow stable and core to its function. And genes move around with, just like our coleslaw that we get with viruses, in bacteria we get things called prophage that move genes. So we try and stay away from genes, if we can, that are associated with prophage. And the last point, step 6, again, is that validation. So once we've identified this target, there is millions of bacterial species we don't even know about. So that's why it's important to do that environmental sampling to make sure we don't get false positives. In this project, we had 6 targets, and don't have to really worry about all those words, except that we wanted to finish off our work with Erwinia amylovera. The way we saw that was it was a gram-negative bacteria and a big priority for Australia. Ratheobacter toxicus was one that the USA, Jim, was really interested in. So it's an endemic bacteria into Australia. It's not present in the USA. Pseudomonas aeruginosa pathophyreactinidae, well, that was when we were writing this, that was about the time with all the heat in New Zealand. Even though Australia's got a very small kiwi fruit industry, we saw that as a risk, but particularly for the US, they really wanted to know what the situation was with that virulent strain. Xanthomonas citrus, or citrus canker, is pretty topical at the moment, but it's actually a gram. It's a really important one for us. Candidatus Liberibacter solanacearum, well, it's a non-culturable bacterium, so it's a bit of a shift from those top four. It's related to zebra chips, so big issue for New Zealand, we don't have it. Again, very topical. So it's also vector-borne, so we started to think about, well, how do insects play a role in moving bacteria around? So from a science point of view, that was interesting. And solanacearum down the bottom, that was really for us in Victoria to use this strategy, this pipeline, to try and identify some specific targets to help our own potato industry in Australia to move seed potatoes and export, but also in Northern Australia there's a few solanacearum or Ralstonia species that are actually exotic as well. So just an example of the work, just one example of all our work, I just wanted to focus then down on the zebra chip pathogen, and this work was done in Grant Smith's lab down in Plant and Food. So these are the key players. There's 6 names up there, but usually for the last 6 years there's at least 4 of them working on zebra chip, on PBCRC funding, but other New Zealand interests as well. And they've done some great work, and this ground circle again represents the genome of solanacearum. A massive amount of work gone into this, and if we look at these little blue and red marks, basically what we're saying is that these are regions of interest. These are regions that are located near mobile elements, but when we get down to the tin tacks, they identified, I think it's 11 regions or 10 or 15, doesn't matter, these red regions in here, and that's where they decided to design their diagnostic to zebra chip. And that data, that information has gone into some primers and a molecular assay for 5 actually that's gone into the National Diagnostic Protocol that's with SFIT at the moment and should be ratified over the next 12 to 18 months. But we shouldn't forget the science that's gone behind that. So at the start of this project, Grant and his team already had collaboration in Europe and we're doing some work on this haplotype C, and that's being published. So they had science, they're building their database, and then through this work, there's a paper in prep now, Thompson et al., which has got this massive data set, probably the biggest in the world, of genomes on solanacearum, and that identified that ring we were just looking at in the slide, identified our markers. And then critically around the validation, the Kiwis are right on the money looking for other Liberibacters. And so another paper that's going to be, I think, just been accepted as a draft genome is out there now. And this is a species of Liberibacter they found in New Zealand in another host. So we're trying to look at that sort of scope and make sure we don't get false positives. The second half of our project was around field diagnostics, and this was, I was really told 3 years ago about how far we would get with LAMP, and we had it front and square. This is Jim thinking big, yeah, let's go for it. But actually, we've made some really significant advances in field diagnostics. And we had 4 targets you can see down the bottom: fire blight, zebra chip, citrus canker, and PSA in kiwi fruit. And so our goal was to use our data, our genomic information, our diagnostic capability, and not have to, and say to industry, look, we think we can give you a very good assay you can do in the field. You don't have to worry all the time, every time, about sending samples to the lab and waiting for us to do the job in our laboratories. And there's a few techniques that strategies we looked at, but I'm going to sort of focus on one that was our dominant assay, and that was what we call LAMP. You may have heard of that, and it's called loop-mediated isothermal amplification. We started looking at over a year ago, just over a decade ago, just after we got out of the back of the van was probably when we started thinking about LAMP. And the things we really liked about it from a practical point of view is in those first stop points. So because it's at one temperature, mobility-wise, you don't need a big battery. So we could have a machine that we could run out in the field for 10 or 12 hours, whereas the machines we use in the lab, this PCR stuff, it cycles. So heat, cool, heat, cool, heat, cool, a lot of energy. So that was a good thing about isothermal from a field perspective. The second thing was the polymerase that actually synthesises the nucleic acid, so it creates our sensitivity, is incredibly stable. And so if we had crude nucleic acid, it could still churn out and amplify. When we charge industry for a PCR assay in the lab, probably 50-60% of our cost is getting good quality nucleic acid. So to be able to not have to worry about that crude nucleic acid in the field, where we could be a lot cheaper and quicker and easier and still get amplification. We know it'll affect sensitivity, but is it good enough? We thought that was worth a look. The actual test takes under 60 minutes, so in the field you want answers now. No use doing it in the field, still takes 2 days. And the sensitivity still stood up really well with PCR, and that 10 to 100 times less is still conservative. People don't like me saying that, so no, it's equal to conventional and only tenfold behind qPCR, and that's true. But I think it's better to be cautious, and because I still think 100 times less than PCR in the field is an incredibly powerful diagnostic. So that's why we thought it was potential, and so we had these regions. We also had to do is design primers. Our real interest was actually taking it to the field, that transition from lab to field. And so this was our strategy. We wanted in-field validation of our smart surveillance tool. We wanted to train end users. We wanted to learn firsthand from them how difficult it was. What's going on there? And we also wanted to identify and implement improvements. So we had this sort of circular thing going for the last 3 years. And just wanted to point out over the right-hand side is this RPA, which is another in-field assay that the Kiwis needed to use to actually amplify Liberibacter solanacearum. Its genomic properties weren't compatible with LAMP technology. So that's just, but for simplicity, I'll just stick with the LAMP. So in-field validation, out we went, just out the back of the ute, at the back of the SUV, and this is a Kiwi orchard in New Zealand. I just wanted to point out, just because Jim's not here to hang it on him, we often call this the stack stance. So whenever he's taking a photo, he's got that huge lens. He won't use his phone or anything. He's sort of got centre of gravity behind him, so the lens does it. Even when he's taking samples from leaves, he's always got both feet firmly planted. It was these gems we really got a lot from Jim. You never knew when they were coming up, but we all really appreciate it. So we optimised as scientists, as technicians in a lab. We went out and thought we knew what we're doing and got some strategies together. But the real fun came when we went to the actual surveillance team. So these guys here are actually biosecurity surveillance staff from Victorian government, and we were under some kiwi plants up in northeast Victoria, and we were testing for the bacterium, Pseudomonas ringi, not so much Actinidae, because we knew it'd be around up there. So we spent a couple of days with these guys, and what we did was we were in under the orchard and we had leaves. We went and sampled, and that was all written down, and we showed them how to do it. And then about 2 or 3 hours later, after talking, we said, right, you go and do it. And so this lady, her name was Bev, and Bev is dead set champion. She is a great surveillance officer, but she's got about as much lab experience as my mum, and that was perfect. So she'd never been in a lab, and a lot of our surveillance staff were exactly like that. So this to us was a true target. And so Bev taught us more than we taught her in that 3 or 4 hours. And some of the things we take for granted, like using a pipette and actually keeping the tip pointing down, was something that she couldn't. She'd start talking and waving it around, and contamination, and we're watching it, and oh God, it was funny. But we knew that we had to, what we thought was simple had to get more basic, and which is great, because that's reality. And so what we ended up with was this 6-step procedure. And the first step was that we had a red cap tube with a ball bearing and the extraction buffer in it. And so that went just to shake. But the really important thing we did was we got rid of pipettes and we standardised the tubes down the bottom. So when we got rid of the pipettes, we just used sterile loops. So all the staff in the field has to do is no measurements. They just have to put the loop in like you're blowing bubbles and transfer it to another tube. We've had great success. We have a lot of other interest outside of the CRC, and you look at that list of organisms. So we've gone from viroid to virus, bacteria, and we're moving into the insects. The fruit fly lamp is an absolute winner. Mark Blackett, Linda Zhang done a lot of work on that, and we've had it out to industry. We've used it in the field. Works a treat. Works on larvae, which is for insects absolute gold. The important blocker, it's not a blocker, the important challenge we've got with LAMP now is that right-hand column around commercial. And this commercial is not so much to make money, it's not even that. Commercial is an avenue, it's a pathway for end users to have access to the technology. We want them to be able to buy kits to do their own surveys. And so we see that as a critical step now. We know LAMP works on all those organisms. Now we've got to make it accessible to Bev. She rings up, orders a catalogue, I need 200 reactions, off she goes and does what she's got to do. So just to finish up, our key outputs, our capacity, we've increased the capacity of plant bacteriology of trained PhDs. Our strategy is generalised genomics-based strategy, and this is for in-lab assays as well as in-field. Our delivery has been really significant and we've smashed these milestones, even if I do say so myself, in that we've produced 3 NDPs and 2 equivalents in the USA, so 5. Put that together with Linda Zhang's PEQ grains and we've produced 9 NTPs from the CRC in the last 3 years. Great work. And science outputs is essentially around manuscripts. We've got 6 published, 9 in preparation. In the last 3 years, we've done 36 talks, and a third of those have been keynotes or plenaries. Our 5 NDPs, which are book chapters, by the way, in Australia, get on board, add to your resume. And with the workshop, with the LAMP technology, we've done 5 workshops and over 120 participants and users. But, whoops, sorry, Barb. What I want to do now is, from an end user's perspective, just Barb to come up and just to summarise her thoughts, not biased, on our project, and I'll just finish off with our recommendations.
Thanks, Fella. I'll make this very quick. Yes, I know I've just got 2 minutes. So for those who don't know me, I'm the outgoing chair of SFID. He's the incoming one, and I'm the one that nags people about getting your protocols verified and up to date and getting them back to me. And from our point of view, as an end user, for the person that uses these protocols to keep ourselves safe in case of incursion, the things that were really critical about this project was the keeping pace with the changes in science. Rapidly developing field of genomics, this is really nailing some of it, and for us it's really critical to keep on top of that so we've got the most accurate identification in the heat of battle. The international collaboration is absolutely vital, and this has been a really good indication of that. We've got to develop these international collaborations because these pests aren't in Australia and you have to work on them in real life. The human capital, we have such a hugely increased value of people now that have the knowledge that can help us in incursions, and that's been shown, the value in the heat of battle with the citrus canker, with the TPP and the CSLO interest. It's been really critical in that so that has huge value as an end user. But the one thing that I will say for all the policy people in the room as well, we really have to keep the policy with pace with these scientific changes. Because those of us who know with the citrus canker, there was a few little issues there, and we really have to try and keep the policy up to date with the science. But it was a great project, great work, fella.
Thanks, Barb. So just to finish off, because Pete's getting really angry with me, and we had 4 recommendations at the end of our report, and one's a no-brainer, and we just got to keep reinforcing it. I know we're on the same page. Continue investment in sciences and human capital to underpin plant biosecurity. Some of the PhDs in plant bacteriology you'll see today and tomorrow and seen over the last 3 years, the science, it's world quality. We want to promote industry development of end-user applicable field detection kits, so we are getting that external investment in LAMP, and we need to get commercial on board, but we're working on that. Interesting, isn't it? It's like that in-field validation. Increased investment in the theory and practice of sampling. It doesn't matter what test we do, we've still got to get the plant or the insect into the tube. And how do you do that? And how do you do that reliably to maximise a quality answer? There's still a lot of work to be done there. And we want to continue supporting international collaboration, as Barb just said. I can't give this to Jim Stack, but this final statement makes a lot of sense to me. I think it's his, but we won't tell him that. My indigenous pathogens are your exotics, and your indigenous pathogens are my exotics, and that's why we've got to look offshore when we do research in biosecurity. And on that, I'd just like to acknowledge those groups and thank you.